Welcome back, students. Today we will continue our discussion in pharmacokinetics, and today's part is metabolism, drug metabolism, also known as biotransformation. Drug metabolism or biotransformation. So again, a review. What's pharmacokinetics? Is the what the body does to the drug. So what the body does to the drugs, and discussed under 4 headings, that is absorption, distribution, metabolism and excretion. Metabolism is also known as biotransformation, and in short they are known as ADME. So our today's discussion is about metabolism. What is biotransformation or metabolism? It is a chemical alteration of a drug in the body. So it is the chemical alteration of the drug in the body. It renders the lipid soluble non-polar compounds to water soluble polar compounds, and it is excreted. So the metabolism is a process which is prior to the excretion. So as we know, our all biological membranes are lipid layers, so we and non-polar, so the drug should be in lipid form and non-polar, then only it will be absorbed. But for excretion, again we have to cross that area, and it should not be reabsorbed. So it should be water soluble and it should be polar. So the biotransformation or metabolism is a process which is a prior procedure before the excretion, and it makes the lipid soluble non-polar compounds water soluble and polar. But the lipid soluble substances will be reabsorbed from the renal and extrarenal excretory sites, which prolongs the duration of action and also leads to toxicity. That's why we have to make the drugs water soluble. So if the drug is lipid soluble, it will be reabsorbed from the area of excretion also. So when we are excreting a lipid soluble drug, it will be absorbed from there also. So it will be reabsorbed from the renal and extrarenal areas. So it can lead to prolongation of duration of action, and also it can lead to toxicity. And the water solubility aids in excretion of drug or metabolites. That means these lipid soluble drugs should be made to a water soluble drugs, otherwise it will be reabsorbed. So we have to get a smooth excretion of the excess drugs or the metabolized drugs. So for that we have to make the drug water soluble, and the metabolism is the process by which the drugs are made as water soluble drugs. Now the main sites or organs of drug metabolism. The primary site is liver. So in our body, the liver is the main organ which metabolizes everything. And for biotransformation, there are some secondary organs also. So liver is the main site, and there are some other organs like kidney, lungs, intestine, skin and brain. Kidney, lungs, intestine, skin and brain. So in detail, before that, the results of metabolism. So what is the fate of the drug after metabolism? First fate is inactivation. So an active drug will be made inactive. Okay, so an active drug can be made into inactive. And another process is it making an active drug to an active metabolite. So the drug itself is active, and the metabolized product is also active. For example, allopurinol. Allopurinol is a drug used in gout, and the metabolite, the first metabolite is alloxanthine. Again, this alloxanthine is also active metabolite. Same way the morphine. Morphine, we know it is a painkiller, or it's like a sedative. The metabolite morphine 6-glucuronide also have the same properties of morphine. So an active drug can the metabolite also can be active, and sometimes the active drug will be made inactive. The majority of cases, more than 90% of cases, an active drug will be made inactive. This is the main process of metabolism. In some cases, the active metabolite or the active drug which metabolizes, and the metabolite is also active. And another case is inactive drug made into active metabolites. Okay, some inactive inert substance, inert drugs after metabolism will become active. Like example, like levodopa. Levodopa will be metabolized to dopamine. So levodopa is a drug used in Parkinsonism. enalapril, and its active metabolite is enalaprilat. So the inactive drug will be made into active drug. Now we coming to the phases of metabolism. The metabolism has got two phases. One is phase 1, which is known as non-synthetic. Simple reactions like oxidation, reduction, hydrolysis, cyclation and decyclations. These all are phase 1 reaction. Then comes the phase 2 reaction. It is a synthetic reaction. So here some substances or some factors are added to the drug or the metabolite. That is known as conjugation. They are conjugated like glucuride, acetylation, methylation, sulfate, glycine, glutathione. These all are added to that substance or added to that drug to make metabolism. In short, when there is a drug, a few amount will be used for our actions and the rest will be unchanged and will be excreted. Some substance will undergo phase 1 reactions, or some drugs will undergo phase 1 reaction and a metabolite is formed. That metabolite, sometimes the first phase 1 metabolite may not be possible to excrete, so it has to undergo phase 2 metabolism. Some majority of cases, phase 1 after phase 1 reaction the metabolites will be excreted. In some cases the phase 1 may not be enough, so it has to undergo, the metabolite has to undergo the phase 2 reaction and the metabolite will be excreted. Okay, so some drugs will undergo phase 1 reaction, the metabolite will excrete. Some other drugs will undergo phase 1 reaction, the metabolite will not excrete, they go to phase 2 reaction, then the metabolites are formed and they excrete. And some drugs, they directly go to phase 2 reaction and the metabolites will be excreted. So the metabolism is a process. The whole metabolism process is a forerunner for the excretion. So all the process, phase 1, phase 2 reactions, all are a prior procedure for excretion. Okay. Now in detail, phase 1, non-synthetic reactions. It is the introduction of a functional group or exposure of a new functional group molecule. They are adding or losing of some substance to a simple substance, simple functional unit into a drug. Polar functional groups like OH, NH2, SH, COH are either added or unmasked if already present on the lipid soluble drug. So the resulting product may undergo phase 2 reaction. So phase 1 reaction is again the phase 1 reaction is a first step for phase 2 reaction. It makes the group like the polar functional group. That means the unpolar drugs will be made polar by adding OH or NH2 or SH or COH, or either if the drug itself having these groups, it will be masked, so the drugs will become polar. Now the first type of phase 1 reaction is the oxidation. Oxidation you have already studied in your high school classes. It is the adding of oxygen, addition of an oxygen or negatively charged radical, or removal of hydrogen or positively charged radical. So it is the addition of oxygen or a negatively charged radical, or removal of hydrogen or positively charged radical. It is catalyzed by microsomal and non-microsomal enzymes. So this process for the oxidation, we need some enzymes which will be located in microsomes or some without or other than microsomes. So in the microsome it will be located in the smooth endoplasmic reticulum. The non-microsomal enzymes will be seen in the cytoplasm and mitochondria. So we need some enzymes. They are microsomal, either microsomal or non-microsomal. Oxidative reaction will form an hydroxylation, dealkylation, oxidative deamination. So these all are the final product of oxidation reaction. The main drugs which will be oxidized are barbiturates, phenytoin, theophylline. So these all are drugs which are metabolized by oxidation reaction. Now a few words about microsomal enzymes. They are located in the smooth endoplasmic reticulum, mainly in the liver, kidney, intestinal mucosa and lungs. So liver is the main organ for metabolism, and the kidney, intestinal mucosa and lungs are also a secondary organ for metabolism. They are inducible, so that means Normally they won't be very active, but when we have some drugs or when these will be produced according to the need. So they will be produced only when there is a need. That means inducible. The catalysis, oxidation, reduction, hydrolysis, glucuronide conjugation. So all the processes, all the phase 1 reactions can be catalyzed by these microsomal enzymes. Example, monooxygenases, cytochrome P450, glucuronide transferase. So these all are different types of microsomal enzymes which will activate or which will catalyze our phase 1 reactions like oxidation, reduction, hydrolysis, glucuronide conjugation and all. Glucuronide conjugation is a phase 2 reaction. Now, non-microsomal enzymes, they are mainly present in the cytoplasm and mitochondria, mainly the organs like liver, plasma and other tissues. It is not inducible, so it cannot be induced, but they will be already present in the body. The reaction catalyzed like hydrolysis, some oxidation and reduction reaction and all conjugation except glucuronidation. So the non-microsomal enzymes are the mediators or the catalyzers. The majority of them will be catalyzing the phase 2 reactions except the glucuronide conjugation. Glucuronide conjugation we have already seen, it will be catalyzed by the microsomal enzymes. And also hydrolysis, one of the hydrolysis reaction in the phase 1 reaction is also mediated by our microsomal enzymes, and some oxidation and reduction also, but the majority of oxidation, hydrolysis, reduction is made by the microsomal enzymes, and with that phase 2 reaction called glucuronide conjugation. Except these glucuronide conjugation, all other conjugations are catalyzed by non-microsomal enzymes. And the example of these enzymes are flavoprotein oxidase, esterases, amidases, conjugases, etc. Now reduction, one of the second metabolism, reduction. It is the converse of oxidation, so it is the opposite of oxidation and involves the CYP450, which working in the opposite direction. So CYP450 is a cytochrome P enzymes which are produced in the liver. So the oxidation reaction, the reduction reaction is a converse of oxidation, so the mere opposite of oxidation reaction, which is done by CYP450 enzyme. So it is the addition of hydrogen to the drug molecule, and it is less frequent than oxidative reaction. The reduction generates polar functional groups like hydroxyl or amino groups, which undergo further metabolism. So it will move to phase 2 reaction. Examples like alcohol, aldehyde, quinolones are reduced. So these all are the drugs, or these are the substances which are reduced in the body, undergo reduction reaction. Then the primarily reduced drugs are chloramphenicol, warfarin and halothane. So these all are very commonly used drugs. Chloramphenicol is a broad spectrum antibiotic, warfarin is an anticoagulant, and halothane is a anesthetic. So these all are the drugs which will undergo reduction reaction. Now hydrolysis. Hydrolysis means no change in the state of oxidation of a substrate, but the drug molecule is cleaved after addition of molecule of water. So hydrolysis means the adding of water. The drug molecule will be braked after addition of a molecule of water. So ester plus H2O, water, gives acid and alcohol. For that, esterase. The catalysis is esterase enzyme. Mainly take place in the liver, kidney, plasma and intestine. So the drugs which undergo hydrolysis are the drugs with ester, either amide hydrolysine linkage. So the drugs who is having one of the end is having an ester or either or a amide or a hydrolysine will undergo hydrolysis. So they will be easily undergone by hydrolysis. So if there is ester at the end of a molecule, then it is easy for a water to add with the esterases. Okay. And the example of these drugs are procaine, lignocaine, aspirin, carbamazepine and oxytocin. So these all are the important drugs which will metabolize by hydrolysis reaction. And some minor pathways. Minor pathways, some phase 1 minor pathways, cyclation and decyclation. Cyclation means formation of a ring structure from a straight chain compound. So the straight chain is made into a ring. And the example is proguanil. Proguanil is an anti-malarial drug. So it is a formation of a cycle or ring from a straight compound. So it is known as cyclation. Decyclation means opening of the ring. So if a ring is formed, it will be opened and make a straight chain compound. Example, barbiturates and phenytoin. So decyclation is also a phase 1 reaction. Now phase 2 or synthetic reactions. Here conjugation of the drug or its phase 1 metabolite with an endogenous substance to form highly polar product which can be effectively excreted. So here is the conjugation, adding of a endogenous substance to a phase 1 metabolite or a drug directly. So the substrate for phase 2 reactions are phase 1 metabolite or the drug directly itself, and the product will be highly polar product, so it will be easily excreted. Endogenous substances for conjugation reaction derived mainly from carbohydrate, amino acids, and possess large molecular size. So the endogenous substances will be derived or formed from carbohydrates or amino acids in the body itself, and they will be having a large size, large molecular size. And the metabolism or the actions are relatively faster than the P450 catalyzed reaction. So the cytochrome P450 reaction will be faster than the mechanisms or the activity by P450 enzymes. And this require high energy and also specific enzymes for specific reactions. The molecular weight of the metabolites is important in determining the route of excretion. So the molecular weight of the metabolites is important in determining the route of excretion. So the final product of already I have sold the substance which are produced from the carbohydrate or amino acids will be having highly polar, and also they will be having high molecular weight or large size. So the size of the metabolite, the final metabolite, determined the route of excretion. If the final product, final metabolite after phase 2 reaction has got high molecular weight, it will be mainly excreted through the bile. And the substance which have low molecular weight after the final metabolism, they will be excreted through urine, because the low molecular, low sized particles cannot be filtered through our Bowman's capsule in the kidney. Large molecular weight or large substance cannot be filtered. That's why it is the low molecular weight substance are excreted through urine, and the high molecular substance are excreted through the bile. So except glucuronation, all other phase 2 reactions are carried out in the cystosol. That means all other reactions are non-microsomal, so it is outside the microsome. As the availability of endogenous conjugate substance is limited, saturation of the process is possible, and unconjugated drugs metabolite may precipitate toxicity. That means, as I've already told, these substrates are formed from the carbohydrate or amino acid in the endogenous substance in our body. So when there is this reaction is a limited process or saturable process. So if there is 100 drug and 100 endogenous substance, which can easily conjugate, but if there is only 50 endogenous substance available and there is 100 drug, the 50 will be left over, it cannot be metabolized. So the unconjugated metabolite, the drug or the metabolite can precipitate in our body itself and causes toxicity. Okay, so it is a saturable process. Now phase 2 reactions, glucuronic acid conjugation. It is the major conjugation reaction. It is catalyzed by UDP glucuronyl transferase. UDP glucuronyl transferase is an enzyme. And the types of substrates are phenol, alcohol, carbolic acid, hydroxylamines, and sulfonamides. And by glucuronate conjugation, it increases the molecular weight of the drug. That means it will be excreted mainly in the bile. Examples are morphine, nitrophenol, acetaminophen, diazepam, digoxin, and hydroxydapsone. And also our some substance in the body, endogenous substance also undergo, or endogenous compounds itself undergo this glucuronate conjugation. Example like bilirubin, steroid, and thyroxine. These all are also metabolized in our body by this glucuronate conjugation. Next one is acetylation. Here the conjugation is with an acetyl group. So the acetylation is done by adding a acetyl group to the compound. So cofactor acetyl CoA, and enzyme is N-acetyl transferase. So we need a acetyl CoA as a cofactor for this action. So all the acetylation reaction will need a cofactor, and the process is done by an enzyme N-acetyl transferase. And here also, in some individuals will be slow acetylators and some individuals will be fast acetylators. So if the individual is slow acetylator, the drugs which are acetylated, or the drugs which are metabolized by acetylation reaction will be slow. So there is a chance for toxicity in that type of people. And also in people who have long or fast acetylators, the drugs which will be metabolized by acetylation reaction will be metabolized very fast, and also it reduces the time or duration of action in sort of people. So the acetylation, there is two type of acetylators. One is slow acetylators, one are fast acetylators. The drugs which are mainly acetylated in the body are sulfonamide, isoniazid, clonazepam and diazepam. The drug isoniazid is a drug used in tuberculosis, treatment of tuberculosis, and the side effect is peripheral neuritis and hepatotoxicity. Here the difference, when the patient is a slow acetylator, it can lead to peripheral neuropathy, and if a patient is a fast acetylator, the toxicity is hepatotoxicity. So both the slow acetylation and fast acetylation is dangerous. Both can cause different toxicity. One is peripheral neuritis and the other one is hepatotoxicity. Next one is clonazepam, which is an important drug used in epilepsy. Again, here also has two type of adverse effect. One is ataxia and the other one is dizziness. Procainamide, which is an important drug used for ventricular tachycardia. Here also two type of reaction, that is SLE and hypotension. Sulfonamide, antibacterial agent, hypersensitivity reaction and hemolytic anemia. Hypersensitivity in fast acetylators and hemolytic anemia in slow acetylators. Hydralazine, again used in hypertension. Hypotension is an adverse effect, tachycardia and headache. So hypotension and tachycardia in a group, and the headache for the other group. Methylation. Next phase 2 reaction is methylation. Here it is the transfer of methyl CH3 group from a cofactor S-adenosine methionine, that is SAM, to a acceptor substrate by methyl transferase. Again, here it is a process of adding some additional endogenous group to a substance. Here it is methyl added with help of methyl transferase with another cofactor needed is S-adenosine methionine, SAM. For acetylation we need acetyl CoA. For methylation we need S-adenyl methionine. The types of substrates are phenol-like compounds, amine substances, catecholamines. The drugs are dopamine, pyrimidine, epinephrine, histamine and thiouracil. So these all are the drugs which are metabolized by methylation. Sulfate conjugation. Here it is the conjugation with a sulfate group from 3-phosphoadenosine 5-phosphosulfate. The enzyme involved is cytosolic sulfotransferases. The drugs like phenol, the substance like phenol, alcohol, aromatic amines are conjugated by sulfate conjugates. So the drugs which having this type of phenol, acetol or aromatic amines are acetaminophen, methyl dopa, estrone is an estrogen compound, 3-hydroxy coumarin. So these all are substances which are, or drugs which are metabolized by sulfate conjugation. Fifth is the glycine conjugation. It is a minor pathway and a type of substrates and drugs with carbolic acid. So the substance or the drugs who we have in carbolic acid at a site will be metabolized by glycine conjugation. And mitochondrial acyl CoA, glycine transferase enzyme. Sorry, the mitochondrial acyl CoA is cofactor needed, and the glycine transferase is the enzyme needed for this process. The drugs like salicylic acid, benzoic acid, nicotinic acid, cholic acid and deoxycholic acid. So here all are acid substances, or the drugs which having carbolic acid at the end. And the last phase 2 reaction is glutathione conjugation. Glutathione conjugate with reactive electrophiles catalyzed by glutathione S transferase. Again it is seen in the cytosolic or mitochondrial or microsomal. Cytosolic glutathione S transferase. Cytosolic glutathione S transferase will metabolizes all the drugs or xenobiotics. Microsomal GST, that is microsomal glutathione S transferase, is an endogenous metabolism of prostaglandins and leukotrienes. So the glutathione reaction can occur in both the cytosolic or microsomal. So the cytosolic reactions are mainly for the drugs and the xenobiotics, that is the external factors which we give to the patient. So but the microsomal, the inside process is for the endogenous substance like prostaglandin and leukotrienes. The types of substrate or the drugs which having epoxides, nitro group, hydroxylamines and aryl amines will be metabolized by glutathione. And the drugs are acetaminophen, bromobenzene, ethacrynic acid. These all are just examples. There are different types or numerous number of drugs which will be metabolized by glutathiones, but these are the important examples. And a few words about first pass metabolism or pre-systemic metabolism. This we have already discussed in the class of absorption. So this is the metabolism of drug before reaching the systemic circulation. So in the initial class I have told the drug should be first absorbed, then it should be distributed to different areas, then it should be metabolized, and finally it should be excreted. So this is a long process from absorption to excretion. So when the drug is moving from absorption to the site of circulation or to the blood circulation, before reaching the blood circulation some metabolisms occur. It is the first pass metabolism. Orally administered drugs exposed to drug metabolism enzymes in intestinal wall. So intestinal first pass metabolism, and also liver, hepatic first pass metabolism. So these two is the main organs which undergo the first pass metabolism when the drug is given orally. Okay. And it can be avoided. How can, if the drug is given orally and it undergoes first pass metabolism from intestine or the liver, so how can we administer to avoid the drug, avoid this type of reaction? We have to use other routes of administration. They are parenteral administration, sublingual or transdermal. Parenteral means we are giving injections. Sublingual means we are giving, or the tablets are placed under the tongue. So in the anatomy class you may be studied about the anatomy of tongue, the bed survey of tongue. So the blood will be, some will be absorbed directly and the others can go, a few amount can directly go to the blood circulation. Okay. And in the transdermal also same mechanism. The blood will be absorbed, the drug will be absorbed from the skin layers directly to the blood vessels. And in the previous class I have told about some induction and inhibition. Some compounds can inhibit some metabolism or can induce some metabolism, or some type of enzymes can be induced or inhibited. So few words about induction of drug metabolism. Xenobiotic activate transcription. This is the induced expression of gene encoding drug metabolizing enzymes. So some substances or some drugs have a property for inducing some metabolized enzymes. I have already told that for a metabolism occurring in the body we need some enzymes, and the microsomal enzymes are inducible. So we have to induce by something, even the product or by some other product. Here some other products will, or some drugs will induces the liver or the microsomes to produce the enzymes which will metabolizes some other drug. So if a drug A is there and some drug B is there, we are administering both. One is an inducing agent A. So the drug A will increases or increase the production of enzymes which need for the metabolism of drug B. So when drug A increases the drug B's metabolism, means it will fastly, the drug B will be fastly metabolized in the body and the action will be slow, or action will be low. So it induces their own metabolism and others. So there is a property of inducing their own metabolism and also others. Consequences and the byproduct will be decreased intensity or duration of action of the drugs. It can lead to tolerance, interference or adjustment of dose of other drugs, metabolism of endogenous substrates. So these all I have mentioned, the substance or metabolism in our body for the drug and also endogenous substances. So when two drugs, and out of that one is inducing agent, means it will induces itself the metabolism of itself, and also it induces the metabolism of other drug or some other endogenous substances. So finally the action of the other drug will be reduced, or duration of action will be less, and there is a chance for tolerance, development of tolerance. And these are some cytochrome enzymes, and the drug metabolize the drugs which are metabolized by that enzyme, and the inducers of that enzyme. So here omeprazole and smoking, it is an inducer of CYP1A2, and the 1A2 is metabolizing the acetaminophen, tamoxifen, theophylline and all. Again, rifampicin, phenobarbital is a drug inducers. So you have to study the names of drug inducers. It is an interaction between two drugs, and one is inducer of the other drug. So you have to study the names of inducing agents like omeprazole, smoking, rifampicin, phenobarbital, cyclophosphamide, barbiturates. Okay, so these all are common drug inducing agents. Next is the counterpart, is the inhibition of drug metabolism. Involvement of the same enzyme or a cofactor sometimes lead to inhibition of the metabolism of one drug by another. So same way the one drug is inducing other drug, metabolism of other drug, same way one drug can inhibit the metabolism of other drug. So if the metabolism of other drug is inhibited, it will be accumulated in the body and leads to toxicity. Metabolically generated reactive intermediate react with the P450 upper protein or heme moiety and irreversibly modify the upper protein. So this is the mechanism. And the drugs like fluoxamine, galamine, clopidogrel, ticlopin, trimethoprim, sulfaphenol, and tiol, these all are the drugs, examples of the drugs which are inhibitors. So they can inhibit the metabolism of some other drugs, inhibit the metabolism of acetaminophen, coumarin, artemisinin and all. So you have to study the names of drug inhibitors, metabolism inhibitors. And as a summary, a drug first it should be absorbed, then it should be distributed, then metabolized and eliminated. So some drug after absorption for metabolism, it undergoes the phase 1 metabolism. Sometimes it will be conjugated, or phase 2 reactions may be occur, and it can directly eliminate. Some drug phase 1 reaction, sorry, I will show here. Here a drug is absorbed and there is no phase 1 reaction. Instead of it directly goes to phase 2 reaction and it is excreted. Some other drug, it undergoes phase 1 reaction. Phase 1 reaction, the drug metabolite with the modified activity, it will be conjugated and excreted. And inactive substance sometimes may need conjugation, will be excreted. Some drug directly can excrete, some drug directly will be excreted. And the lipophilic drug have to be made hydrophilic. Here you can see the lipophilic, for absorption it should be lipophilic, so it should be friendly to lipids. But for excretion the drug should be hydrophilic, it should be friendly with the water, and only it will be easily excreted. So this is the summary of the metabolism, and you have to study different phases of metabolism and different types of phase 1 reaction and also type 2 reaction. And any other important point in this chapter is the induction and inhibition, the microsomal enzyme induction and also inhibition. So you have to be thorough about the examples of induction and inhibition. So for exam, there will be questions related to inhibition and also induction. And for that we can stop here. Drug metabolism, and one more class is there about excretion. So we will be discussing in the next class about excretion. And thank you once again, students. Stay home, stay safe. Regards, Dr. Rahul. Thank you.